Hello and welcome back. We will now proceed to talk about semantic segmentation using deep neural networks for this week, and that will be the last of the lectures for this week. And for next week, we'll talk about generative models. Okay. We've all seen this kind of a segmentation that we introduced in one of the earlier weeks where we used a contour to initialize, and then the contour slowly converged around the object of interest. So very clearly there was a foreground and a background object, as is seen here, where lots of little circles have been initialized, and over time they all converge to the object of interest. Okay, so this is the kind of segmentation that we've all worked with. Okay, so with deep neural networks, the segmentation takes the process is different, and we will look at what that is. So let's consider this Lxnet architecture. Okay, it's typically used for image recognition. So one of the earliest papers to do semantic segmentation, which is basically labeling every pixel in the input image as belonging to a set of classes, one class or the other. Okay, so we know that the image recognition networks, just like this Lxnet over here, typically give you one output per class, which means that let me clarify what I mean. What I'm saying is that if you're trying to classify an object as belonging to one of 1000 classes, then the output layer typically has 1000 output units. Okay, each of them is a value which corresponds to probability of belonging to that class. Okay, but what we want instead is this map shown here, which is pretty much the same size as the input, and with every pixel given a class label or probability of belonging to a class, and that is, if there are three classes, then you will have three such images in the output, with each image corresponding to probability of a certain class. So we'll explain more of that in the later slides. Okay, so the idea is now to take a object recognition based network and use it for image segmentation, wherein every pixel of the input image is labeled as belonging to one class or the other. Of course, the number of classes being provided depending on the data. Okay, so how does that work? So let's just look at this Lxnet architecture. So we know that, for instance, this 256, right? So basically this layer here, typically Lxnet is of size 7 cross 7 cross 256, and it's fully connected to the next layer, which has 4096 hidden units. Okay, so the idea here to get to fully convolutional neural network is to convert all the fully connected layers into convolutional layers. So how will we convert fully connected layers to convolutional layers? So in this case, if we take this layer, it actually has 256 feature maps. Each feature map is of size 7 cross 7. So then if we define 4096 filters, okay, each of size 7 cross 7, but then they'll automatically run across the channel dimension. So each filter is actually of size 7 cross 7 cross 256, and if we have 4096 filters, then the output of this is the filter size. If this filter acts on this input, then the output is of size 1 cross 1. Okay, so your output would be 1 cross 1 cross 4096. This is your output. Okay, so then, okay, so now that we have done this, what is the advantage of doing this? Okay, so we know that in the case of a image recognition, the input size was approximately 224 cross 224. Okay, now if that input goes through this typical Lxnet, the output is a vector of size 1000. Okay, and each element indicating the probability of belonging to that class. Okay. Now, if we take this, let's say let's consider this input image, which I'm going to say is twice the size of this image. Okay, twice the size of the image that you typically give as input to this image recognition network. So you would have, instead of 224, if this was 224 cross 224, your input would then be 448 cross 448. Now, if you have converted the network to fully convolutional neural nets, fully convolutional layers, then instead of 496 cross 496 cross 1, since I have doubled the size of the input, then your output eventually, if we use these convolutions properly, then your output would be 1000 cross, I should say, write it another way, 4 cross, 2 cross 2 cross 1000. Okay. So the output would be maps of size 2 cross 2, 1000 of them. Okay. And these 2 cross 2 indicate, so if we can split, so I'm going to do this very crudely. It's a 2 cross 2 feature map at the output. There are 1000 such feature maps, and each of these feature maps would indicate the probability of a particular class, let's say cat or dog. So very easy way of understanding where does this 2 cross 2 come from is that we split this input into 2 cross 2, then the probability of this, so if we have, let's say, 1000, there is the first two are corresponding to cat and dog. Let's look at the first feature, 2 cross 2 feature map. It will say whether this big superpixel block here is cat or dog probability. I shouldn't say cat or dog probability. Let's say the first feature map is cat. Then this particular block, what is the probability of it being cat? This particular block, what is the probability of that being cat? So on and so forth. And if you go to the second feature map, let's say it's corresponding to the class dog, then it will do the same thing, except that now the probability values would indicate that of being a dog. Okay. Now, one clear problem we have seen is that, this is a very coarse feature map. Okay. Coarse feature map. So, but we would like the probability for each pixel as belonging to a particular class, which is what gives you the segmentation map, correct? So then the way to do that would be to upsample this. Okay, so this upsample process is often mistakenly called deconvolution, of course, but the better thing would be to call it transposed convolutions. Okay, or strided convolutions or fractionally strided convolutions. Transposed convolutions would make sense. We'll see what those are over the next few slides. But what you do is basically you take this 2 cross 2 and upsample it to the size of the image in order to get a fairly high resolution map. Okay, so how would you go about doing that, right? So I'm just giving you in this paper what they did. So they had this crude this rule of thumb. So if you look at the Lxnet, okay, if you have an input of size 224 cross 224, okay, you would, if you come to this 256, this size would become, after a few layers, this becomes, the size is 7 cross 7. There are, of course, 256 channel, and this is 224 cross 224, let's say. Okay, so there's a factor of 32 reduction, so it becomes 1 over 32. Okay, so that's typically what this is indicated here. So the idea is, if you have a input of a certain size, by the time it comes around here, let's say even in this particular layer, you have it's been downsampled by a factor of 32. So this is a typical architecture, right? So these numbers tell you the size. So let me zoom in a little bit to show you. So this is 3D is 3 channels of certain size, and then if you do 2 convolution layers, non-linearity followed by max pooling, you would end up with 1/2 the size, 60, and of course you can have as many outputs as there are filters, which in this case is 64. Similarly, you do another set of convolutions, max pooling, you lose another factor of 2 subsampling, so your size is 1/4. Then you double the number of feature maps. Similarly, 1/2 the size again, double the number of feature maps. You do that till you get to 1 over 32. Okay. So this is the typical, loss in resolution size as you go through the a neural network, a convolutional neural network from the input to the output side. Because this paper came out in 2015, so they were using some of the best performing networks on the image recognition challenge, which was basically Lxnet, VGG, etc. Okay, and all of them had this typical format wherein if you come to if after a certain number of layers towards the end of the towards the output layer, your size is about 1 over 32. Okay, so then the idea is how do we get very high quality upsampled segmentation maps. So before we go any further, I just wanted to show you, what would the loss function, how would you calculate the loss function? The loss function is calculated per pixel. So you would do this, the equivalent of this binary cross entropy, which is y log y, or categorical cross entropy, if you want to call it. And this is computed per pixel. So how would you interpret it? So your ground truth will tell you what category that particular pixel is. Okay. So your ground truth will have, so let's suppose you're working on in this case, a 4-class problem. Okay. 4-class problem. 4 classes, boat, person, cat and dog. Okay. So let's say your ground truth, let's say the object you're looking for in that image of the 4 classes, either the pixels corresponding to dog. You have to write down the pixels corresponding to dog, right? So let's say this, I'm just going to take the example of 1 pixel. This we know for sure is dog. The others, or if that is dog, then it can't be anything else. So cat is 0, person is 0. So this is the ground truth. With the label 1 correspond to this pixel here, says that pixel belongs to dog. Your output, like I said, will have as many feature maps as there are classes. So in this case, there are 4 classes, there are 4 feature maps, and each feature map, the pixel values in each feature map is the probability of that corresponding class. Okay. So we say this is a boat class. So each of the pixel values will tell you the probability of that pixel belonging to boat. Okay. Similarly, for the person, cat and dog. So you would use these to calculate your pixel-wise loss functions. That's typically what you would do. And of course, this summation is over the classes. For every pixel, you will have the categorical cross entropy over the classes. And you would you will take the average loss over the entire image. Okay. So, but this is how you would interpret the output. You will have as many feature maps as there are labels, classes, and the feature map, the pixel values correspond to the probability of that pixel belonging to that class. Okay, so then how would you do this? Some more classes. So if you look at this pixel, then you take this out as a vector of outputs, and this would be a vector of your targets. Okay, and you would use that to compute this pixel-wise categorical cross entropy. Okay, once you have that for every pixel, you would do that for all the pixels, and then you would sum over for the entire image. Okay, so y here is a 4 vector, y hat here is also a 4 vector. Okay, that's the idea. So this is how you would calculate the loss function for semantic segmentation. So now that you have converted all the fully connected layers to convolutional layers, so all of them are convolutional layers now, but however we saw that even with that architecture, the final layer, the output feature maps are very coarse in a sense. It's a factor of 32. Size reduction is a factor of 32 because of the sequence of convolution and pooling layers. Okay, so one way that this problem was solved in that paper that I was referring to is by using the intermediate layers also to provide predictions. Okay, so for instance, pooling 5 layer after upsampling can be used to provide an 32x upsampled prediction. Okay, now you can take the pooling 5 layer, do a 2x upsampling. You can take the pooling 4 layer. Of course, once you 2x upsample it, pool 4 are about the same size as pool 5 once you upsample this by 2x, because every time this pooling layer is just a factor of 2 reduction in size. Okay. So then between two successive pooling layers, you will have a factor of 2 upsampling. So pool 5 and pool 4 you can combine, okay, and then you can 16x upsample it to provide a prediction, okay. And of course you can keep doing that, you can sum them and use that. Similarly, you can take that summation, upsample it one more time, take the third pooling layer, combine that and provide one more upsample prediction, okay. So basically, of course I urge you to go through the paper as well as follow these upsamplings properly, but the idea is you take intermediate outputs, okay, from the network and use that to provide high resolution information in the decision layers, okay. So that you get a decent high resolution segmentation map, okay. So this is one of the earlier techniques for fully convolutional networks. Now, when I say upsampling, okay, I'm taking this pool 5 layer or pool, let's say here, say this is factor of 2, this is a factor of 4, let's say this is factor of 8, and 16 factor, then here is factor of 32, reduction in size. How do I upsample it? So what is this upsampling? How is this done? Okay. Now this upsampling is accomplished by what we call transposed convolutions. Okay. So why is it called a transposed convolution? We will look at it briefly. So again, it is also sometimes referred to as deconvolution, which is technically not right, because deconvolution is used in signal processing literature in a different sense. So let's not use that. But of course, in the deep learning community, deconvolution filters are, I guess, acceptable. But transposed convolutions are fractionally strided convolutions, okay, are used, okay. And there's two ways to do this. One is you can just do some high resolution upsampling, like interpolation. Okay, you can do spline interpolation, cubic spline interpolation and all that, B-spline interpolation and all that. And that you do not have to learn the parameters. Okay. However, if you do this transposed convolution, etc., you will learn them. Subsequently, they'll be slightly better than just using splines or linear interpolation or anything of that sort. Okay. So why is it called transposed convolution, etc.? We will see. Okay, so we're going to be looking at this transposed convolutions. Where does it get the name from and how we do that? Okay, so let's say your input feature map is of size 3 cross 3. Okay, this is your input feature map, call it X, and then you have your filter kernel, which is your weight matrix W, which is of size 2. So this is 3 cross 3. It's 2 cross 2. Your output will be 2 cross 2. Okay, this is your Y output. Okay, so if you do a cross correlation of X with W, XW would give you Y as the output. So the idea is now is we want to upsample. So what do we have to do? What is this operation which, when applied in this direction, will give rise to the X? So the way you would do this one is you will superimpose this matrix here, okay, and that will give you one, and then of course you can do this every time, give you the other one. Similarly, you will superimpose it here, give you the third and fourth, so on. Okay. The idea, the cited convolutions work slightly differently. So what you would do is you would wait. So let's say we take this Y, which is typically what we do, and we want to turn this into 3 cross 3. So you will take every element here. Let's say I have 0, 1, 2, 3. Okay. And so your strided convolution, I will also denote that by your transposed convolution. I will also denote like with this symbol, with let's say the same kind of 2 cross 2 filter would be to multiply this every element of this input with the filter. Okay, so you're going to weight the filter with this element. So that will give you, in this case, let me redraw this. Your output will look like this, right? So you will actually end up with a 3 cross 3. Okay, this is not a good diagram. Okay, so let me draw this for one element. So I'm not going to give you the elements itself, but your output in this case will turn out to be 3 cross 3. If you do it, the way to do that is we have this 3 cross 3 output. But the way we would construct it is we will take this element in the input, multiply each one of the elements here with it. So then, of course, this will all be zeros. So then you will get, in this case, you will get 4 zeros. Okay, and then plus you make one more same size. Okay, now we are going to be multiplying with 1 with each one of these elements. So, but you will put it here, the output here. So 1 multiply with this, 1 multiply with that, 1 multiply with this and that goes directly here. Similarly, you will have, I'll do just one more just to show what's happening, and you have this here, right? If you just draw, for instance, so you take 2, multiply this element, this element, this and this, all these 4. So you will get 4 numbers here. Similarly, you take 3 and do, and then you would add them all up. Now, see that there are these overlap regions where you just sum them up. Okay, overlap regions where you sum them up. So this is with a stride of 1. If you increase the stride, then you will get a slightly larger output feature map. Okay, here is 3 cross 3, then you will get 4 cross 4. Okay, so you can have one way of looking at this. Why is it called a transposed convolution? So if you can think of the using x and w to get y, think of it as a matrix multiplication, vector matrix multiplication. So x is a vector, w is a weight matrix. You have to write this 2 cross 2 a kernel in the form of a weight matrix so that you can operate it on x, which you will treat like a vector. Then your output is y. And of course, when you go the other way, that is, you're going this direction from y towards x, then it's basically equivalent to multiplying your input y with a transpose of w. Okay, so you can, that's one way of understanding what is going on internally. Okay, so that's why it's called a transposed convolution, but however, the actual computations are done this way. Okay, so having said that, there are of course many other ways of getting a high resolution output segmentation map. So one other technique which is used is the encoder decoder architecture. Okay, so again, all of these have their origins in the computer vision community. So all the inputs are typically either grayscale images, 2D grayscale images, or RGB images with 3 channels. Okay, so it's very simple, similar to a convolutional network. You have an input, you have a bunch of convolutional layers. Okay, and of course convolution followed by max pooling. But every time you do a max pooling, you would remember the indices which survived. Like in sense, right? If you have 2 cross 2 max pooling, you would take only the maximum out of that. So other 3 elements are gone, but you would remember the index of the one element that you kept. Okay. So as you go from left to right, this piece is called the encoder. This is called the decoder. What the encoder does is do a succession of convolution and max pooling operators till it comes to a certain size, and where after this point, what you do is you would start doing the upsampling. The way you would do the upsampling is you will try to mirror the encoder side. So on the decoder side, you will initialize your feature map with the inputs in the larger size feature map, and you'll only populate those locations from which you acquired the max pooling outputs. Okay, so let's see what that means. And then you do the convolution, and for every stage, every time you want to increase the resolution, you would once again populate that larger size feature map based on the indices that you had from the encoding layer. So let's look at slightly more detail. Okay, so there are no fully convoluted layers, again, connected layers, all of them are convolutional. A decoder will upsample the input using the transferred pixel indices, transferred pool indices from its encoder to produce a fast feature map. Then you would perform convolutions with a trainable filter bank to make it more dense. Okay. So it's only, remember, we're only copying the indices, not the value. Okay. So let's say, for instance, here it is. I'll just zoom in. So for instance, if you look at this particular output, this is the decoder size output. Let's see the decoder. So decoder outputs, on the decoder size, the feature map, first feature map we get is ABCD. Okay. So now you want to map to a slightly higher filter kernel. So from the corresponding stage in the, let's say that you are here and you want to go to this size. So you would go to this stage, find out the indices from which the max pooling operator took the values to get to the lower size feature maps. And then you would put these elements into those indices. Okay. And then now you would do convolutions on this. This is typical 3x3, 4x4 convolutions depending on size of your filter kernels. Okay. So you would do as many convolutions as you would like. And then once again, you would, let's say, from here to there, if you want to go from here to there, then let's say you will take the inputs, they take this feature map, okay, find the values here. Of course, you have a list of values there. And since you want to go to a higher resolution feature map here, let's say I use a different color, say higher resolution feature map here, you would go back to the same level there, okay, find out which indices were taken to get to the lower resolution feature map, and then populate the values there only in those indices, and then continue to do convolutions. Okay, that's a typical way. That's one of the encoder decoder networks that was in vogue for some. Okay, of course, when it comes to biomedical image segmentation, okay, the most commonly used network architecture is the Untitled architecture. Okay, so this is again like a workhorse of biomedical image segmentation. Pretty much, if you use any kind of architecture is accommodated here. So we saw a bunch of architectures that were for image classification, right? Object detection, etc. This is Lxnet, VGGNet, we saw ResNet, and then DenseNet. So all of these can be accommodated in these layers, as long as you can have again, this also has an encoding and a decoding layer, except the one advantage it has is it also has the skip connection, so called, indicated by the gray arrows, which will transfer the feature maps and concatenate them in the decoder side. Okay, so that's what gives rise to that Untitled architecture thing. Okay, so I'll just follow briefly through the encoder side, a couple of maps and a couple of maps on the decoder side, just to give you an idea of how this works. Okay, so here we'll zoom in a bit just to see clearly. So I'll just, so that you can see one or two of these layers. Okay, so this was used for first microscopy images. So the image is slightly larger than what you might have seen. So the first image, of course, these are grayscale images. You can also use RGB images. Just these filter kernels will change to accommodate all the channels. Okay, so the input image tile is 572 cross 572. Okay, then you would use 3x3 convolutions if you like. Okay, that will give you again valid convolutions. That's why you have a decrease in the size. Okay, so because 572 minus 3 plus 1 will give you 570. So a couple of such things. And then this dotted region is basically you would crop it. Okay, you would crop it, and that crop is what is concatenated there, but we will not worry about that. So after a couple of convolution layers where you have 64 filters each, you have a downsampling layer. This downsampling is typically by a factor of 2, and then of course a succession. So this is the downsampling here, and then we have 2 successive 3x3 convolutions, 64 filters each. Once again, you will have, after you downsample, you have a couple of 3x3 convolutions, 128 filters. The same sequence as you had seen before. You would have, you would increase the number of filters, but the size of your feature maps would decrease, by either downsampling every time. So once you do that, you would come to this part, the U, where that U happens. Okay, just move that. All right. So here, you have once again the red arrows indicate downsampling, right? So every time you have a red arrow, it's a downsampling by factor of 2. And the blue arrows indicates convolutions, it's 3 cross 3. Of course, the non-linearity I always never mention, but it's implicit. Non-linearity is of course there, okay? And so once you come to a certain point, you would just, in this case, you will see, if it comes to this 512 feature maps, 64 squared, you have a downsampling 32 squared. The size of the feature map again, okay, is 32 squared. Then you have a convolution, 3 cross 3 convolution, get you 30 squared, 1024 of them. And then you have a 28 squared feature size feature map here. So that is one pathway. The other pathway goes from 512 feature maps, 64 squared to, there is, you just copy, this is where you do the copy and crop, okay. So you have copied and cropped a 56 squared area from here and concatenated it with the feature map from this pathway, so to get about 1024 feature maps, okay. So then in this way, this is the upsampling pathway, okay. So you have a sequence once again of convolutions, and then there is a up convolution once again, okay. So this up convolution can be your shredded convolutions to get, in this case, from 52 to 104. Now, if you see that why we have this cropping operation, as we come to this layer, the corresponding encoding layer, this filter size maps or the feature maps are big, so you would crop them to the size that is appropriate. So, for instance, here, once you go from 52 upsample, you go 104 squared feature maps. But here it's 136 squared, so you would crop 104 squared and concatenate. Once again do convolution and upsample, here you would crop and concatenate, so on and so forth, okay. So this is a typical pathway, and all the way to the top, where in the end you will have an output segmentation map. Here once again there are as many segmentation maps in the output, the final layer, as there are classes, okay. So let me move that to see. In this case, for this problem where they are demonstrating in this paper, two classes, and foreground and background. So there will be a one feature map output, which is the probability of foreground, another feature map probability of background, okay, or two classes, whichever way we want to look at it, okay. So this is the typical architecture for all unit related things. So in the sense that your input again can be of any dimension. So we are going to look at 3D short while. So and then followed by a sequence of convolutions, downsampling, etc. And then at this U layer, this part where it's flattened out, that's when you would start to crop and concatenate on the way up. So the cropping and concatenation from the encoding to decoding layer is just to provide information for better upsampling and better prediction. Okay, so that's it, because a lot of the high resolution information is there in the encoding pathway, which is lost when you try to decode. So you need to give that input in order for better decoding. Okay, so that's like thousands of variants of this seemingly, okay, and of this 3D unit. I haven't shown you 3D unit based, but typically, this is one input channel here. There could be multiple input channels, not necessarily one input channel. So that's where the 3D comes from. We'll look at that later. But the general idea is you have this unit families, right? There is a skip connection. I'll just point out here, skip connections, always encoder block and a decoder, okay? And these blocks are basically can be any kind of sequence of convolutions, upsampling, downsampling, batch norm, dense net. So for example, each block can be a dense net block. Each block can be a ResNet block. Each here, then I say block. There the computations can be ResNet like or densenet like or ResNext. There are so many others, so many different types of architectures. They can all be plugged in here as modules. And of course, the idea is as you go down, you are losing resolution. As you go up, you are building resolution. Of course, the skip connections themselves, they have variants of it. So you can put multiple layers here. You can have, a bunch of layers, convolutional layers here, or you can just, copy and copy, or you can do a bunch of 1x1 convolutions to combine across feature maps. So all of these are possibilities when you do the skip connection also. So this is the general abstraction for the various UNet-based architectures that are out there. Some variation of this is typically employed to get better results, depending on the problem. Okay, so we are just going to look at the so-called 3D UNet. Okay, so it's basically the 3D counterpart of your 2D UNet that we have looked at so far. So what do you mean by 2D UNet? 2D UNet is basically your convolutions are 2D. But of course, we have seen that even in 2D convolutions, your filter extends across the channel. Okay. So when we see 2D UNet, the input is 2D. Okay. There's only one channel typically, and that's a input. And even if there are three channels, each channel is two-dimensional. Okay. That would be the best explanation for 2D inputs. Right. So for instance, RGB image has three channels, but it's still a 2D problem because each channel is two-dimensional. Okay. But for 3D UNet, it can have multiple channels, but each channel itself is three-dimensional. Okay. So this particular paper, which I have cited here, which describes this 3D UNet, describes a situation where there are three channels, but each channel is a 3D volume. Okay. So it's not just a 2D. Each channel is not 2D at the input side. Okay. Then your convolution kernels are three-dimensional. So instead of a 3x3 kernel, you would define, I would say 3x3x3 kernels. Okay. And the interesting part is that this, of course, will extend across, I'll put it here, channels. Okay. So this becomes a tensor. It becomes difficult to roll this in your head. But that's the idea behind the 3D UNet. Everything else remains the same. Okay. Now you have to understand that when you do the first set of convolutions, your convolutional kernel, again subsequently in all other convolutions, this convolutional kernel extends across the channels, where each channel is 3D. Okay. So that's the idea. The channels themselves are three-dimensional. Your basic filter kernel is 3D. And of course, all the operations remain the same. Everything else remains the same. Of course, the idea behind doing that is that, a lot of the medical images make sense when they are viewed as 3D objects. So for instance, when you are looking at an organ like a heart, or even if you are looking at a tumor, a pathology, in this case a tumor, you would notice that the organ is a 3D structure. So if you view successive cuts through the organ, which is what we call slices, we remember our earlier classes about how we can make 3D volumes from CT or MR imaging. The idea is the successive slices, 2D slices in a volume are correlated, so it's best to view them as 3D structures rather than 2D structures. So when you do the correlation, when you do the convolution, and the convolution kernels have to be 3D, because this so-called the translation invariance, the stationarity applies in 3D sense here. Okay, but of course it's complicated. The computations are complicated by the presence of extra channels in the input, each channel having have a 3D form. Okay. So once again, this paper was used to analyze microscopy images, but this is basically sectional microscopy, where you have multiple sections in an image, and they showed that they had very few images, but they were able to get good results. Okay. So once again, I urge you to read the paper, but this is the general idea. Now there are all these software packages like Torch and TensorFlow, which have predefined 3D convolutional kernels that you can use like modules, and if you want to do a rapid prototyping. Okay, so we have looked at a couple of architectures only. I showed you the encoder decoder architecture. We also looked at UNet. UNet is the most commonly used architecture, or there are variants of it everywhere. But that's the most commonly used. Of course, you would incorporate everything in this architecture. For instance, there are transformers incorporated into this architecture, etc., called TransUNet. There are different variations of this, like I mentioned. The skip connections can have their own convolutional layers, and so on and so forth. So this is again just a brief overview of semantic segmentation. But in medical image analysis, this is like the most common task. Okay, so if for all pathologies you need segmentation, organ segmentation, etc., and for population studies. So this is a very common task, and there are lots of research papers on this subject. What I've shown you is like one of the basic architectures that are commonly used. Okay, and you can have to build on top of that. Okay, and of course, I haven't mentioned many other recent developments in semantic segmentation architectures or even image recognition architectures, and they have been again incorporated into the image recognition architectures have been incorporated into segmentation architectures, etc. Different tricks, different ways to combine high resolution, low resolution feature maps. All of that is there, but I urge you to read the literature to keep track of that. But the general principle is the same. You have an encoder decoder kind of a situation always, and how you make those pathways are exact is it determines how good your outputs in general are. Okay, so this concludes our week in terms of semantic segmentation or volumetric segmentation, if you want to call it, for medical image analysis. I briefly touched upon some of the architectures that are commonly used. You will also have a tutorial to attend to, which will show some basic processing techniques in the context of deep learning. Thank you.